Assalamualaikum and a very good morning to Madam Ernie and my fellow friends. So today, me and my friend will be presenting a poster entitled Genetic and Immunological Basis of Human African Trypanosomiasis. Trypanosoma brucei-rhodesiense and Trypanosoma brucei-gambiense are responsible for acute and chronic African trypanomyosis respectively. The reservoir for Trypanosoma brucei-rhodesiense are cattle and occasionally human, meanwhile Trypanosoma brucei-gambiense is human. Tsetse fly transmit the African trypanomyosis. To protect the host immune from the host immune recognition, the African trypanosome have developed antigenic variation through periodic changes of variant surface glycoprotein, which is VSG, that cover the whole cell surface. The infection triggered the immunity to mount a robust inflammatory response due to the carbohydrates component of VSG. The adaptive host immunity involves antibodies that are directed against the VSG epitopes. Furthermore, the apolipoprotein L1, which is APOL1, is a primate specific trypanolytic factor that provides efficient human innate immunity. Trypanolysis of tryposome is caused by APOL1 trypanolytic activity. The components of trypanolytic activity consist of trypanosome lytic factor 1 and trypanosome lytic factor 2. So TLF1 is an association of APOL1 with haptoglobin-related protein. TLF2 is an association of the LF1 with IgMs that recognize the pathogen signatures and accumulate upon the infection. Basically, APOL1 moves through the endocytic system. It then generates endolysosome ionic holes due to the acidic pH-dependent membrane insertion. Subsequent transfer of APOL1 containing membranes to mitochondria induce the membrane permeability and release the mitochondrial endonucleus to the nucleus, which then leads to trypanolysis. Next, I'll pass the presentation to Afiqah.
Generally, there are two possible modes of resistance to APOL1, either APOL1 inactivations or resistant to APOL1 activity. So in the case of the Trypanosoma brucei-rhodesiense, the specific serum resistant associated protein, or known as SRA, binds to the C-terminal helix of APOL1 and thereby inhibit the APOL1 pore-performing activity. And interestingly is SRA is derived from the VSG that acquire the ability to interact with the APOL1 through the truncation of its surface exposure domain, and this resulting to the localizations to the endocytic compartment instead of their cell surface. The SRA gene is transcribed by the highly processive RNA polymerase 1 rather than the usual RNA polymerase 2, and this indicates the higher SRA expression required for the TB rhodesiense resistance to the APOL1. While for the resistance of Trypanosoma brucei-gambiense to APOL1 is involved the VSG-derived protein, or known as Trypanosoma gambiense specific glycoprotein, TGS-GP. It stiffens the endosomal membrane where APOL1 should normally insert during its journey within the tryposome. Therefore, it's hindering the APOL1 pore-performing activity. However, in the absence of the hemoglobin, HbHb, such as usually in the hypoheptoglobulinemic serum, TGS-GP-mediated protections cannot resist the high uptake of APOL1. And thus, since we know that human adaptation to malaria is linked more frequent in hypoheptoglobulinemia. So the adaptation of TB gambiense begin by limiting the APOL1 uptake through the L2-10S mutation in TB-HBr, in which reduce the affinity for the ligands. Thus, we can say that the TB Gambiaceae is more likely to respond to a human adaptation to the another parasites, Plasmodium, illustrate an unexpected dialogue between the parasite through the host. The APOL1 arm race. The direct interaction of SRA with the C-terminal helix of APOL1, the destruction of this helix by the mutagenesis allow the APOL1 to escape neutralization by the SRA and enable this APOL1 C-terminal variant to kill the TB rhodesiense. And interestingly, the ability of the C-terminal APOL1 variant to kill the TB rhodesiense were found to be naturally common among the Western African. By the term of G1 and also G2 that are respectively characterized by the two separate mutation and a double amino acid deletion in the APOL1 C-terminal helix, and this lead the resistance to African trypanosomes except for TB Gambiaceae among African society. However, the resistance to the sleeping sickness is a propensity to the development of chronic kidney disease, and this due to the G1 and G2 mechanism. This is because a recent report stated that the overexpression of APOL1 lead to the cytotoxic, while the unfolding of a intracellular isoform of the G1 and also G2 variants, it's triggered the interaction with the APOL3, and this result to the disease formation. So from the figure, we can see that the inactivation of the APOL3 mediated stimulation of phosphatidylinosytol 4-phosphate or PL4P synthesis by the Golgi kinase PL4KB, while the reduction of the PL4P level in G1, G2 expressing podocytes will account for the increased mobility and reduced autophagy that characterized by the podocytes of kidney disease patient. And this interestingly is that a nonsense variant of APOL3 was found to exhibit positive selection in African population, while also causing the nephropathy like the APOL1, G1 and G2 variants. And this does make that because of its inability to be secreted, APOL3 cannot kill bloodstream trypanosomes despite its intrinsic lytic potential. The selective advantage of APOL3 could result from the loss of APOL3 mediated neutralization of the trypanolytic APOL1 variant.
Hi, I'm FAIZURRIN and I'll be talking about the early parasite defenses, which are the cures of motility and TBKIFC1. In skin, neutrophils are rapidly recruited to the site of infection. This facilitates the development of trypanosome colonization. Trypanosomes resist the neutrophil-mediated engulfment and elimination. This activity is related to the parasite motility following ddNase-mediated disruption of neutrophil extracellular DNA traps. Trypanosomes that lack in propulsive motility cannot infect mice even though they have efficient clearance of antibody VSG complex. Therefore, reduction of rate of antibody VSG clearance in motile trypanosomes can prevent an infection. Fast clearance results from lateral mobility of glycosyl phosphatidylinositol anchored VSG. This is related to the high plasma membrane fluidity by the cholesterol trafficking activity of TBKIFC1 kinesin. Therefore, we can conclude that fast antibody clearance is absolutely required for trypanosome infectivity, and the essential variance factor for this activity is TBKIFC1. Now, what causes the TLF2 internalization? It is caused by the requirement of TBKIFC1 for clearance of IgM VSG complexes. This leads to trypanosome killing because TLF2 IgMs bind to the VSG. Therefore, also occurs in TLF1 associated APOL1 uptake, which is linked to the capture of growth factor. Now, TNF2-associated APOL1 uptake depends on a mechanism. This mechanism is intended to help trypanosome survival. Now, I'll be talking about the parasite armory against innate community, which is the parasite release factor. Trypanosome triggers the rapid synthesis of inflammatory components such as TNF-alpha and nitric oxide. Cytokine, firstly, is important for parasite control at the early stage of infection. However, trypanosome growth is unaffected even though there is considerable amount of induction of TNF-alpha synthesis. This is because only membrane-bound TNF-alpha affects the parasite. Multiple receptor-like adenylate cyclase, or ACEs, is known now to counteract the inflammatory response in the early stages. Now, the internalization of AC containing the trypanosome membranes into macrophages, which happens either from phagocytosis of entire parasites or released extracellular vesicles. This internalization of AC promotes the cyclic AMP, which reduces the TNF-alpha synthesis. How it reduces? It reduces through activation of protein kinase A within the macrophages. Therefore, lysed trypanosomes released from the surface of live trypanosomes can condition macrophages to stop synthesizing TNF-alpha early in infection. Also very similar to TBKHC1, which can condition myeloid cells to synthesize anti-inflammatory cytokine IL-10, which then stimulates the growth-promoting activity of arginase 1, which inhibits the anti-parasite activity of nitric oxide synthase. Furthermore, trypanosomes secrete metabolites that modulate host responses. This is important within the central nervous system. Other than that, trypanosomes also secrete aromatic ketose acids such as indole-pyruvate. Indole-pyruvate inhibit the production of pro-inflammatory cytokine IL-1 beta by macrophages. These metabolites suppress IL-6 and TNF-alpha production by glial cells, which are potent inducers of heme-oxygenase 1. Now, the effects from this is that it can give pro-inflammatory or anti-inflammatory responses to lessen the systemic pathologies associated with the infection. So therefore, now we know that the long-lasting infection is required for this transfer of trypanosomes between hosts, given that the mode of transmission is through sex fly, which is quite inefficient. Thank you.
That were used by the parasite against the adaptive immunity for the variant surface glycoprotein variation is that the parasite can escape antibody by the VSG sequence variation that results from a total or partial gene replacement and variable post-translational VSG modifications like O-glycosylation. And meanwhile, the antigenic variations require monoallelic expression of VSG from several hundred VSG genes. Only one is expressed at one time. So this requirement is ensured by the existence of only one VSG expression site body in the nucleus, which can be maintained by the factors such as histone variants or the telomere binding protein RAP1 and its binding component. So the discovery of new protein complex responsible for allelic exclusion containing the chromatin VSG exclusion 1 VX1 protein and VX2, which is an ortholog of the nonsense mediated decay helicase UPF1, is presumed the most significant breakthroughs in this field. So by changing the selected VSG expression site to another in the ES body is a mechanism that will trigger the antigenic variation that results from the DNA damage frequently observed in the VSG expression site due to its open chromatin configuration and high transcription rate. So importantly, the mechanism linking DNA damage to VSG expression site switching was found to involve ATR, which is a DNA damage signaling protein kinase. So the ATR loss was found to alter the localization of the both RNA polymerase 1 and VX1. Furthermore, a putative translation DNA polymerase was able to bypass telomeric DNA damage termed by TBPOI1IE, was also found to be involved in the control of antigenic variations. In addition to VSG expression site switching, DNA combination, such as gene conversion occurring between the active VSG gene and any other silent VSG gene, can trigger the antigenic variation. For the variation and shielding of surface receptor, the best studied growth factor receptors in trypanosome bloodstream forms those from transferrin, which is responsible for iron uptake, and Hb hemoglobin for the heme uptake. So all these receptors have a 3 helical fold that allows them to have an elongate conformation suitable with the densely packed VSG code. So N-glycosylation or a structural kink are thought to limit molecular crowding of the receptors by VSGs and facilitate ligand access to the binding site. The receptor affinities and concentrations of circulating ligand as well as receptor saturation may render the exposed binding region inaccessible to antibody. So since the transferrin is expressed by a group of different VSG-like genes found in various VSG's expression site, the antigenic variation of this receptor occurs when the VSG expression sites are switched. So according to recent studies, immunological avoidance is the primary reason for the N-glycosylation variation. According to another studies showing that the impact of polymorphism surface exposed sites of ligand affinity, it was concluded that variable regions are the most exposed to immune detection, while the N-linked glycans can help shield a large part of the membrane distal region that is not involved in ligand binding.
In conclusion, African trypanosomes have developed multiple mechanisms to counteract the host defense, beginning of the infections by combination of serum motility and membrane fluidity to avoid immediate elimination. There are extreme level of involvement of African trypanosome system, where the extensive antigenic variation allows the long-lasting chronic infection via the necessity activity of many factors ensuring both monoallelic VSG expression and continuous changes of selectively activated VSG gene. Despite the system complexity and identification of several key actors and the precise identification of their mode of action and the long-standing issue of how parasite receptor from macromolecules can interact with their ligands without being vulnerable to antibody attack has been identified. Lastly, in the case of human infection, the trypanolytic factor APOL1 on how parasites and hosts can entertain a continuous arms race with price to pay on both sides has revealed.